Introduction. What can you see in this picture?
We can see a dog and its puppy.
Correct. Can the seeds in this picture produce the corresponding mango plant?
No. Mango plant can only be produced from mango tree.
You're right. Who looks similar to your father? You or your cousin?
I will look more similar to my father.
Do you know what the reason behind this similarity is?
No.
This phenomenon of production of similar offsprings depends on the inheritance of various genes. Objectives. At the end of this lesson, you'll be able to understand Mendel's law of inheritance, understand inheritance pattern of one gene, identify various laws of inheritance, understand Punnett square and its calculation, identify a test cross. Mendelism. Mendel is known as the father of genetics. He experimented on 7 varieties of peas, Pisum sativum. His work was not accepted because of certain reasons initially, but in 1900, his work was rediscovered by Hugo de Vries, Carl Correns, and Erich von Skammerk. Seven traits of Pisum sativum. The 7 pairs of contrasting characters of Pisum sativum chosen by Mendel for his experiment. Why Pisum sativum? Mendel chose pea plant for his experiment for the following reasons. Many varieties of pea plant were available with alternative forms for traits. Pea plants are self-pollinating, so pure lines can be easily obtained. Pea plants can be easily grown. Pea plants have a short lifespan. Its flowers are bisexual. Stages of Mendel's experiments. Mendel's experiments were carried out in 3 stages. True breeding, to find out whether or not the 7 characters noted by him passed on from parents to offsprings. Mendel raised seeds by self-pollination from each of 7 types of plants. Mendel named each of these as pure character. Hybridization of pure plants. Cross-pollination of 2 different varieties of plants produced hybrid plants constituting F1 generation. Self-pollination in hybrids. Each F1 offspring was self-pollinated to produce F2 generation. Inheritance of one gene. Monohybrid cross between tall and dwarf varieties of pea plants is as follows. Results of monohybrid cross. Gene. Mendel observed that some stable factor was passed down unchanged from parents to offsprings. These factors are known as genes. Allele. Genes which code for a pair of contrasting traits are known as alleles. Homozygous organism. Organisms with same pairs of allele. Heterozygous organisms. Organisms with different pairs of allele. Results of monohybrid cross. Dominant gene, the allele which is expressed. Recessive gene, the gene which is not expressed or expressed in the absence of a dominant gene. Genotype, genetic constitution for concerned expression of a character. Phenotype, the apparent expression seen due to combination of 2 genes. Segregation of characteristics. When gametes are produced by meiosis, the alleles of parental pair segregate and only one allele is transmitted to a gamete. This segregation of allele is a random process, and so there is a 50% chance of a gamete containing either allele. Punnett square. Punnett square is a graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. For example, TT male cross TT female is an internal F1 cross. Place one set of alleles from one parent on top, the other set from the other parent on the side to form a Punnett square as follows. Calculating Punnett square. Determine the allelic composition of the progeny merely by adding the alleles together as shown in the following example. Such a cross would result in following F2 generation. 3 tall plants and 1 dwarf plant, 3:1 phenotypic ratio. 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf plant, 1:2:1 genotypic ratio. Test cross. The genotype of a progeny can be found by a progeny with its breeding recessive type. Such a cross is known as test cross. For example, a phenotype of tall plant can have a genotype TT or T small t. Its genotype can be determined in the following manner. In the first case, all the plants are tall, thus the genotype of the test plant is TT. In the second case, tall and dwarf plants are produced in the ratio of 1:1. Thus, genotype of test plant is T small t. Laws of inheritance. Mendel's principles or laws of inheritance are: Law of dominance. This law states that one factor in a pair expresses itself and prevents expression of the other in F1 generation. Law of segregation. This law states that the factors of each character segregate during gamete formation so that each gamete receives only one factor and is always pure. The two factors and traits occur with equal frequency in male and female gametes. Incomplete dominance, codominance, and multiple alleles. Incomplete dominance. It occurs when neither of the alleles for a certain trait is dominant. Thus, an intermediate expression is found in heterozygous individuals. Codominance. This occurs when both the alleles for a trait are dominant and both are expressed in heterozygous individual. Such a phenomenon can be observed with multiple alleles. Multiple alleles. This refers to the presence of 3 alleles for one trait. Example: human blood has 3 alleles, A, B, O. Dihybrid cross. A cross between two organisms considering 2 genes at different loci at the same time is called a dihybrid cross as shown in the diagram. Result of dihybrid cross. A dihybrid cross produces 4 types of plants in F2 generation in the phenotypic ratio 9:3:3:1. 2 new combinations are produced, round green and wrinkled yellow. Inheritance of one character was independent of the inheritance of other character. Law of independent assortment. Law of independent assortment. Factors or alleles of different characters located in different pairs of homologous chromosomes are independent of one another in their segregation during gamete formation and their coming together into the offspring by fertilization. Did you know the application of Mendel's laws to the study of plants' genes are applied to produce dedicated energy crops critical for second-generation biofuels such as varieties of soybean products? Summary. Let us summarize what we have learned. Mendel was the first to study this phenomenon systematically. Mendel proposed that the factors later named as genes regulating the characters are found in pairs known as alleles. He observed that the expression of the characters in the offspring follow a definite pattern in different first generations, F1, second, F2, and so on. The recessive characters are only expressed in homozygous conditions. Phenotypic ratio expressed in dihybrid cross is 9:3:3:1. Three laws of inheritance are proposed: law of dominance, segregation, and independent assortment. Human blood group has three alleles for one trait: A, B, O. Incomplete dominance occurs when neither of the alleles for a certain trait is dominant.
Introduction. What are genes? Gene is a unit of heredity that is transferred from parents to offsprings to transfer information for the expression of certain characteristics. Correct. Can you identify the structure shown in this picture? It is a chromosome. You are right. We know that gene affects the expression of certain traits. Similarly, chromosomes also affect the inheritance of a character. Objectives. At the end of this lesson, you will be able to understand chromosomal theory of inheritance, differentiate between behavior pattern of genes and chromosomes, understand the phenomenon of linkage, understand the phenomenon of recombination. Reasons for non-recognition of Mendel's work by 1900. Mendel's work remained unrecognized till 1900 because his work was not widely publicized because of lack of communication. His concept of genes as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other was not accepted as an explanation for the continuous variation seen in nature. Mendel's mathematical approach was new and unacceptable. Mendel could not provide any physical evidence for the existence of genes. Discovery of chromosomes. In 1900, chromosomes were identified as colored bodies in the nucleus that were capable to double them and divide just before cell division. As a result, de Vries, Correns, and von Schirmack rediscovered Mendel's work. In 1902, chromosomal movement was recognized during meiosis. Genes and chromosomes. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. The behavior of chromosomes run parallel to the behavior of genes. Both genes and chromosomes segregate during gamete formation to transmit only one of each pair to the gamete. The segregation of a pair of gene or chromosomes occurs independently of each other. Independent assortment of chromosomes. The two chromosomes pair aligns independently of each other during anaphase 1 of meiosis, as shown in the diagrammatic representation. Chromosomal theory of inheritance. Sutton and Boveri postulated independently in 1902, the chromosomal theory of inheritance. The theory states that genetic information is carried by the genes that are part of chromosomes. Sutton and Boveri stated that pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors, that is genes, that they carried. Experimental verification of chromosomal theory of inheritance. Experimental verification of chromosomal theory of inheritance was done by Thomas Hunt Morgan on fruit flies, that is Drosophila melanogaster. Why Drosophila melanogaster? They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. There was a clear differentiation of the sexes. Its hereditary variation could be seen with low-power microscopes. Linkage. Linkage refers to the tendency of alleles that are physically close to one another on the same chromosome to stay together during meiosis. Linked genes do not show independent assortment. Morgan used a 2-point test cross to detect linkage between yellow-bodied, white-eyed females and brown-bodied, red-eyed males. He observed that the two genes did not segregate independently and F2 phenotypic ratio was different from the expected 9:3:3:1 ratio. Morgan observed that eye color in Drosophila was sex-linked as shown in the diagram. Crossing over. Crossing over refers to the physical exchange of chromosomes between the homologous chromosomes. It occurs in the prophase of meiosis 1. Crossing over and genetic recombination. Crossing over results in recombination of genetic information between homologous pairs. Recombination refers to the genetic assortment of alleles on same chromosome during meiosis. Linked genes show lesser recombination frequency because these genes do not assort independently. The distance between two linked genes decide whether they will show crossing over and in what frequency. Genetic mapping. Linkage maps correspond to the arrangement of genes on a chromosome. Recombination frequency between gene pairs on the same chromosome can be used to measure the distance between genes and map their positions on the chromosomes. Genetic map distance is equal to number of recombinant gametes upon total gametes into 100. Genetic map distance is measured in centimorgans. Did you know? Linkage map can be used to study the mechanism of insecticide resistance to pests Heliothis and Helicoverpa. Linkage map determines the number and identity of linkage group contributing to resistance to a particular insecticide in a particular strain. Summary. Let us summarize what we have learned. Sutton and Boveri postulated independently in 1902 the chromosomal theory of inheritance. The theory states that genetic information is carried by the genes that are part of chromosomes. Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosomes. These genes were called as linked genes. Crossing over results in recombination of genetic information between homologous pairs. Linkage maps corresponded to arrangement of genes on a chromosome. Introduction. Hey, today I am very happy because my aunt gave birth a baby girl. That's great. But my uncle was not happy because he wanted a baby boy. he was blaming himself for this. But why he was doing that? Actually, a mother is not only responsible for giving birth of a female child, but it depends on the father's gene that actually decides the sex of a child. Let me explain you this mechanism in detail. Objectives. At the end of this lesson, you will be able to understand the mechanism of sex determination, understand mutation, understand pedigree analysis, identify various types of genetic disorders. Sex determination. Chromosomes can be classified into two types. They are sex chromosomes and autosomes. Sex chromosomes. A chromosome involved in determining the sex of an organism, usually designated as X or Y. Autosomes. All the pairs of chromosomes other than the sex chromosomes are called autosomes. Sex determination mechanisms can be classified into three types: exotype, XY type, ZW type. Exotype. In this, males have only one X chromosome, while females have a pair of X chromosomes. Example, grasshopper. XY type. In this, males have only one X and one Y chromosome, while females have a pair of X chromosomes. Example, human being. ZW type. In this, females have only one Z and one W chromosome, while males have a pair of Z chromosomes. Example, birds. Sex determination in humans. Sex determination mechanism in human beings is XY type. Human beings contain 23 pairs of chromosomes, 22 pairs of autosomes, and one pair of sex chromosomes, XX in females and XY in males. During fertilization of an ovum with sperm, two types of zygotes can be formed. Therefore, there are 50% of birth of a male XY or female child XX. Thus, the genetic makeup of the father's sperm is responsible for sex determination and not the mother. Mutations. Variations can be found in progeny due to: number 1, recombinations after crossing over of chromosomes. Number 2, mutations. Mutation. Mutation is a phenomena which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. Mutagen. Factors that induce mutations are called mutagens. Mutagens can be chemicals such as alkylating agents, radiations such as UV rays, ionizing radiations, etc. Virus. Physical agents. Types of mutations. Mutations can be classified into the following types. Deletion. Deletions remove one or more nucleotides from the DNA. Insertion or duplication. Insertions add one or more extra nucleotides into the DNA. Point mutations. These are the mutations arising due to the change in a single base pair of DNA. Frame shift mutation. In this, one or more bases are inserted or deleted. As a result, the DNA sequences become meaningless. Pedigree analysis. Pedigree analysis. An analysis of traits in several generations of a family is known as pedigree analysis. Pedigree. It is a diagram of family relationships that uses symbols to represent people and lines to represent genetic relationships. Some basic symbols used in a pedigree are: genetic disorders. A genetic disorder is a condition that is caused by an abnormality in individual's DNA. Mendelian disorders. a genetic disease showing a Mendelian pattern of inheritance and caused by a single mutation in the structure of DNA is known as Mendelian disorder. Such a disease trait can be dominant or recessive. Pedigree analysis can be used to determine the transmission of such traits. Mendelian disorders. Some of the most common Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria. Hemophilia. It is a sex-linked recessive disorder that impairs the body's ability to control blood coagulation. It is caused due to the deficiency of certain clotting factors. The transmission of this trait occurs from carrier females to unaffected male progeny. The possibility of a female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. The following chart shows the transmission of hemophilia. Cystic fibrosis. Cystic fibrosis is a genetic disease caused by deletion of the gene coding for the protein CFTR, which is located on the long arm of chromosome 7. The loss of usually 3 nucleotides results in changes in sequence and conformation of the protein CFTR, which makes it dysfunctional. The protein CFTR controls the transport of chloride ions across the cell surface membrane. It damages the electrolyte balance of the organism and results in change in exocrine glands. The membranes lining the lungs produce abnormally viscous mucus, resulting in frequent infections and breathing difficulties. The disease also leads to enzymatic failure of the pancreas. This in turn causes disorder in the digestion and absorption of food. Long-term retention of secretion in these glands results in fibrosis of the secretory organs such as the liver and pancreas. Secretory dysfunction causes the sweat gland to produce sweat containing increased concentration of sodium chloride. This is referred to as salty sweat. Sickle cell anemia. This is an autosomal recessive trait transmitted from parents to the offspring when both the partners are carrier for the gene. The disease is controlled by a single pair of allele, HBA and HBS, only homozygous individuals, HBS. The defect is caused by the substitution of glutamic acid bivaline at the 6th position of the beta chain of hemoglobin. Thus, the shape of RBCs changes to elongated sickle cell structure. Phenylketonuria. It is an autosomal recessive disorder that occurs due to accumulation of phenylalanine in the brain resulting in mental retardation. Chromosomal disorders. These are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Such disorders can be aneuploidy. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes, called aneuploidy. Polyploidy. Polyploidy is a condition that arises when cytokinesis does not occur after telophase, resulting in increase in a whole set of chromosomes. Trisomy. An extra copy of a chromosome is called trisomy. Monosomy. Absence of one chromosome from a pair is called monosomy. Chromosomal disorders. Some common chromosomal disorders in human beings are Down's syndrome. It occurs due to the trisomy of chromosome number 21. Klinefelter's syndrome. It occurs due to the presence of extra copy of X chromosome in human sperms XXY. Turner's syndrome. It occurs due to the absence of X chromosome in human ovum XO. Did you know? Donase
Alpha.
is a drug prepared by co-transformation technique by a US biotechnology firm. It aids in the treatment of cystic fibrosis, a Mendelian genetic disorder. Summary. Let us summarize what we have learnt. Sex determination mechanism is of three types: XO, XY and ZW. Human beings follow XY type of sex determination. Mutation is defined as change in the genetic material. A genetic disease showing a Mendelian pattern of inheritance and caused by a single mutation in the structure of DNA is known as Mendelian disorder. Example: sickle cell anemia, hemophilia, etc. Down syndrome occurs due to trisomy of chromosome 21. Klinefelter's syndrome carries an extra X chromosome, while Turner's syndrome lacks a X chromosome.